Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 114th seminar, and I call it on ARPA-H Day. It's been almost 13 months or 2.5 years since I first started this seminar series on August 26, 2021. So far, 333 speakers have signed up for this series, which may last until February next year. Among them, we have one Nobel laureate, Professor Francis Arnold, 21 National Academy members, 22 funding agency directors, including today's pioneer speaker, 20 editors-in-chief, and 9 Cell or Nature Journal editors, as well as 161 rising stars and more than 1,000 global audiences. I want to thank all of you, and to celebrate this achievement and continue to make big impacts together, we'll have the first international conference from December 9 to December 12 this year, 2024, in Hawaii. Why not? It's paradise, I believe. Please stay tuned for further information regarding this conference. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Ravi Basavappa. He is a senior advisor at ARPA-H, a new funding agency focusing on innovative health-related projects. He has been helping to establish a new agency devoted to revolutionary advances in health and healthcare, shaping the future with better health technologies. As a person who had suffered from many years of medical issues or dying years, and who has seen many family members and friends suffering from many medical problems, I passionately hope we'll have a better world with better medical solutions to many problems. Ravi, thank you so much for your great contributions to this important task and also communities, and the virtual podium is all yours now. Thank you.
Thank you very much, Taisuk, for that introduction, and thank you very much for inviting me to participate in this really impressive seminar series that you assembled. It's an honor to be invited. So I will just provide a very brief introduction to myself and then talk a bit about ARPA-H. As Taisuk said, it's a new funding agency. So my background is in biophysics and structural biology. I did the usual PhD, postdoc, and then I took the academic route. Initially, I was an associate professor at the University of Rochester, director of the graduate program in biophysics at NIH funding. So I was pretty well established. But then I acted on my feeling that this really is the golden age of biology. There's so much cool stuff happening across the globe in biomedical research, apart from what was happening in my own laboratory. And my laboratory at the time focused on structural analysis of proteins that were involved in controlling cell cycle division. So it's a fascinating field, but there's so much other really cool stuff happening. And to be able to get that broader perspective, I shifted to NIH to get that broader scientific perspective. And to maybe have a broader scientific impact. For the 12 years before I joined ARPA-H, I ran the high-risk, high-reward research program for NIH, the Pioneer Awards, the New Innovator Awards, Transformative Research Awards, and such. The Wacky Ideas program for NIH. And that was a very cool gig because I got to see such a broad perspective, since whatever was in the broad mission of NIH was could be supported through the program. And then I joined ARPA-H in October of 2022, and full-time, because ARPA-H is like that high-risk, high-reward research program, but it's 100x what that program is able to do. So at ARPA-H, I'm senior advisor. I'm helping to establish the agency in any way that I can. As Taisuk said, it's a relatively young agency. It'll be 2 years old in March next month. And so the ARPA-H uses the DARPA model. ARPA-H, I should expand the acronym. ARPA-H is Advanced Research Projects Agency for Health, ARPA-H. And like its name suggests, it's based on the business model of DARPA. I think most of you have heard of DARPA, the Defense Advanced Research Projects Agency. It has a legendary legacy in the sort of projects that it's funded very early on, being able to recognize tremendous opportunities. Examples of outcomes of DARPA projects are the internet, right? It started as ARPANET, but now the internet, GPS, stealth technologies, and the mRNA vaccines. They were first supported by DARPA and then had a tremendous impact during the pandemic. So ARPA-H, like DARPA, seeks to support really high-risk, high-reward research, things that are that other funding agencies such as NIH cannot fund because of the risk tolerance. ARPA-H not only tolerates risk, but we embrace risk. So we're going after those really blue sky projects, things that are just beyond the feasible, or maybe they don't violate more than one law of physics. It's one way to say it. But if successful, would have a tremendous impact. So that is the space that we're trying to carve out relative to the other funding agencies. So ARPA-H is a pure funding agency. We don't have our own laboratories, but it's like DARPA, it's driven by the program manager model. So program managers are these scientific visionaries. They join ARPA-H having identified a seemingly intractable, but a very important problem. And then through the resources provided by ARPA-H, they then assemble a team of performers. Performers can come from, on the outside of ARPA-H, they can come from academia, from industry, or from nonprofits, wherever the talent is. And then through ARPA-H funds, the program manager supports this team to pursue the vision of the program manager. So the ARPA-H structure is very bottom up. It's driven by the program managers. The scientific portfolio of whatever we're supporting at a given time is determined by the particular cohort of program managers we happen to have at that time. So opportunities for you to engage with ARPA-H, one would be to be a program manager. So and don't be misled by the title program manager. This is really a highly enabled position, empowered position. You would have you would have available to you many resources provided within ARPA-H, as well as tremendous resources to really pursue your vision. We have a lot of information on our website, so I encourage you to check us out there, ARPA-H.gov. Another way to engage with ARPA-H is if you want funding for your own projects. We have what's called an open BAA, Open Broad Agency Announcement. So through that, you can submit projects or proposals that are relevant to the broad mission of ARPA-H. They don't need to be aligned with any particular program manager or program. Anything that's relevant to the broad mission of ARPA-H is welcome. I should warn you, though, that the receipt deadline for ARPA-H, for the open BAA, is March 14th. So you have a bit of time, but I encourage you to check that opportunity out as well. And you can also look at the programs that are made public now, and if there are any opportunities through the program manager created programs, then you would be able to submit proposals through that mechanism as well. But I'll stop there. Just think about ARPA-H, be willing to take risks, and And I will turn it over to Taisuk. Thank you very much.
Thank you so much for very informative and important message to all the community. You know, actually, I interact with ARPA-H when ARPA-H started, including Rene, the director, entire director of the agency. And I even consider myself as a potentially a program manager. And then I realized I probably need to have more experience. I'm also still love to do research myself. So probably later on, I may consider again to become a program manager. But I highly encourage other people who listen to this talk, just consider, because this job basically making huge impact to the community and potentially the health, area, and then potentially helping a lot of patients. So why not? I mean, you creating huge impact, what else do you want to do or achieve in your life? So even later on, I decide not to pursue this time for the program manager. I converted my recommendation as a paper, perspective paper. I published one paper about ARPA-H and recommendation to ARPA-H. So I'm very honored to have you today. Basically, you giving, short but very important message to the community. And then thank you so much for your time today.
Thank you, Taisuk. And I encourage you to continue thinking about being a program manager. We'll have a continuing need for program managers.
Yeah, I'll definitely consider.
Good,
yeah.
good.
Thank
you
you.
so much.
Okay.
Unfortunately, I need to leave, but thank you.
Yeah, thank you so much. I mean, keep in touch.
Yeah.
All right. Okay, now the main speaker of today with a longer introduction. Dr. Donghoon Lee is a rising star in biology programming, very unique field, I believe. He has very interesting career path until now, and I will, introduce him more in detail. He began on his journey in the field of just traditional biology at the University of Toronto, earning both his bachelor and PhD in cell and systems biology. His doctoral research centered on the role of small GTPase in regulating the membrane cytoskeleton during cell division. He undertook a postdoctoral fellowship in Dr. Elizabeth Chan's lab, initially located at Johns Hopkins, but later moved to the University of Texas Southwestern Medical Center, or UT Southwestern. His significant contributions were recognized with a prestigious 3-year postdoctoral fellowship award from the Canadian Institute of Health Research. During this period, he crossed paths with Dr. Dae-hwan Kim, an assistant professor at UT Southwestern, who ignited his passion for biology programming. Dr. Kim's vision that the construction of biological systems bears a striking resemblance to software development, implying that life could be programmed similarly to how software is programmed by software engineers through computer programming languages, resonate deeply with Dr. Lee. This encounter marked a turning point in his career, and after completing his postdoc, he joined the Professor Kim's lab to finally pursue his long-standing interest in computer programming and synthetic biology. Currently, he is developing a novel computer language, L++, designed specifically for crafting biosystems biology and other life science applications. He will share probably more insight about this groundbreaking work with us today. I was so fascinated by this idea and concept. Actually, when I visited them to give a seminar, UT Southwestern, and I strongly believe that L++ is set to instigate the paradigm shift in the way we approach biomedical research and applications. Donghoon, thank you so much for your great contribution to this great, exciting effort, and please take it away. It's all yours now.
Thank you for a generous introduction, Dr. Moon. Let me share my screen first before I begin.
Good.
Looks like it's there.
Yep.
Okay, hello everybody. I'm Moon from Dr. Daewon Kim's lab at UT Southwestern. Again, thanks to Dr. Moon and the SynBYSS community for this chance to discuss our work on biology programming language. Today, I'll speak to you about the need for a biology-specific language and introduce L++, discuss the Human Code project, and explore L++'s relevance in this era of AI. So first, why do we need language for biology? Well, our scientific approach in synthetic biology especially involves designing and testing biological systems, aiming for efficiency. Yet modeling these systems remain a challenge. Unlike electronics and computer software, where they have dedicated languages to streamline their system design, biology lacks an optimized language for system design and modeling. Specifically, within electronics, popular hardware description languages like Verilog, VHDL allow for precise circuit design by controlling the data flow, behavioral and structural information of digital circuits, exactly the things they need to handle in their system. And similarly, computer software, their development utilizes various programming languages, and these languages enable the manipulation of data and the construction of algorithms and logic sequences that processing units will need to execute. Again, exactly the things that they need to handle is provided by the language. Interestingly, biological systems bear striking similarities to computer software in their structure and functionality. For instance, an operating system interact with users and hardwares through a series of hierarchically organized components and modular devices within it, designed to perform specialized distinct tests. And biological systems engage with their surroundings, responding to various stimuli such as temperature or the presence of nutrients, and managing tasks like toxin elimination and communication with other organisms. So these systems are also hierarchically structured, with organs performing specific autonomous functions. And their modular organization, rather than reliance on a single essential component for all biological functions, enables the adaptability and have allowed evolution of biological entities to happen over time. So we can also compare how both computer software and biological systems are represented. Computer software operates on binary, but software engineers today don't really use this language directly to design their program, but rather use high-level programming languages for a more intuitive development process, moving beyond direct binary manipulation. Similarly, biological systems operate on a genetic code, sequences of DNA that are not inherently interpretable or convenient for human manipulation. This necessitates a high-level biology-specific programming language that abstracts cellular language, making it accessible and modifiable for us. Thus, in the same way, computer software is crafted and modified using programming languages. biological systems could be engineered using a customized biology programming language. And this would allow us for the translation of live code into even simulation as another type of compiler output, enabling virtual experimentation and analysis of this biosystem. There are several specialized biology programming languages exist for describing aspects of biological systems, certain aspects of biological systems. Notable examples include BioNetGen and Kappa languages, and BioPeppa and Cello for biochemical network analyses and molecular interaction studies, process algebraic analysis, and genetic circuit design, respectively. Yet these languages often adopt syntax and features from traditional hardware description and computer programming languages, and this approach may lack intuitiveness and flexibility for biological applications, facing challenges in scalability and model support. Alternatively, computational models of biological systems can be directly developed using traditional software programming languages, as demonstrated by a series of seminal research projects that created wholesale models for well-characterized prokaryotic systems like Mycoplasma genitalium, E. coli, and the minimal cell. While these models are invaluable in the community and a lot of research, their development itself and application are complex for most biologists to use. And that's because it requires expertise across multiple disciplines to generate this type of product. Like that includes software engineering skills, simulation biology, chemistry, physics, and mathematics. All of these understanding all of these disciplines. And typically that makes it a multi-author, multi-year projects, making them challenging to undertake for those who are looking to apply such models within their own projects. So it looks like a truly comprehensive, intuitive language for representing modeling biological systems does not yet exist. And we're talking about the one that allows biologists to easily construct and scale their models for computational simulations to test hypotheses. A suitable biology language should enable the description of both chemical and biological components, detailing their identity, quantity, location, timing of introduction or removal in the system, and functional roles across the various scales from atoms to ecosystem. This implies that the language should facilitate the description of processes in a manner that does not require exhaustive details about every molecular detail and be able to handle the unknown processes reasonably. And with such a language, if it's available, in essence, biology programming would mean that it would utilize live code written in such a language that's completely decoupled from the backend computational directives. So one doesn't have to deal with computer programming aspect itself, software programming itself. And the language compiler would transform it into a simulation module. And that should allow biology programmer to execute and view and interact with the simulation, effectively bringing virtual life into existence. It's virtually yet physical and functional aspects of the system within this biometaverse. So, what is ARPA-H? Like Tessa, Dr. Moon has introduced in the earlier, Dr. Daewon Kim initiated a project aimed at creating a programming language tailored for biology, which was later named ARPA-H. The core design principle behind ARPA-H was to develop an intuitive language for biologists, one that could be mastered in no more than a day. That's a good goal to have. ARPA-H incorporates biology-specific syntax to achieve that for handling basic and complex chemical names, units, and their spatiotemporal dynamics, multi-resolution, dynamics. And additionally, L++ integrates traditional programming syntax and data structures to improve code efficiency and allows high-level description of incompletely understood processes. So let's get started and dive into what it looks like to use L++. First of all, starting with a visualization of a hydrogen atom. The most intuitive representation for a hydrogen atom we thought might be H. The symbol of hydrogen equals 1, indicating a single hydrogen atom. And when you run this code, we should get a hydrogen atom appearing. And it won't be doing anything because it just says there's one hydrogen atom. In fact, simply using just the letter H should be sufficient for us to say there is a hydrogen atom. Otherwise, I would put it right there. Assuming this hydrogen is already defined, or any objects that are previously defined by this point, we should be able to instantiate that to see the object as an output. And this can streamline the expression a little more. L++ differentiates between integer and floating point numbers. So here, 1 and 10 integer, they mean an absolute quantity of this object. But when you say that with floating point numbers, this represents a continuous values. That is molar concentration L++. You can either omit it or include it. It recognizes also conventional chemical names, and which may include numbers and special characters and even chemical formulas. And creating new objects is straightforward. For instance, we can specify a type of a new object by using the name of a previously defined object, in this case, a keyword cell, and give it a name, C in this case, which can be elaborated on with a code block enclosed in curly braces below. Note that C here, in this code at least, is no longer carbon as we wrote that. And within this, it's possible to include, within this curly braces, we're able to include various entities, including water, molecules, and such. And furthermore, we can adapt the properties. We can adjust the properties of existing objects as well, like their shape and dimensions. Using the keyword petri dish here, we can instantiate a petri dish without needing to assign a unique name to it. I just wanted a petri dish. Doesn't have to have a name, although custom names are an option. And we can make it a 15 centimeter diameter petri dish and populate it with cells by simply including these C in the curly brace here. And additionally, we can name individual cells. So you would see something like this as an output. And you can also give some cells a special names for tracking purposes, perhaps, maybe tag 1 and tag 2, and even be able to control where they are relative to the petri dish dimension with this parentheses next to the name of the object, like this. And tag 1 would appear right here, tag 2 will appear over there. And the same language applies, same rules for proteins. We can represent a globular protein, designated as P1 here in this manner. And by default, the visualization will display a globular actin monomer. To depict a protein with a specific structure, you can also assign its PDB ID, access the PDB property of P2 protein here, and give that a PDB ID. In this case, that's GFP, and you should be able to see the GFP volume. Furthermore, constructing a protein complex is straightforward. You can just simply link two molecules with a tilde operator between them, and you can also assign the name for it as well. In this case, P3, and you can instantiate that. You should also be able to see a linked molecules of P1 and P2 together, that should show up as 3. How about reactions and pathways? Reaction objects contain chemical equations and reaction parameters, and they can be grouped into a pathway. So for example, in our bacterial chemotaxis model, in this pathway, we utilize mass action reactions with both forward and reverse kinetic constants alongside Michaelis-Menten kinetics, describing these two enzymatic reactions, two sets of enzymatic reactions, and they depict the binding of chemoattractant to the receptors and the associated regulatory modifications. And importantly, the concentration of the ligand down here is controlled over time, indicated by specifying a sequence of time points in square brackets following the ligands. And that's the quantity for that type of control. The simulation data may be explored and analyzed through diverse types of data visualization techniques. And integration of L++ code, right? We don't want to write everything in a single file for a large system. Integration of L++ code is also convenient. In this code, see how E. coli contains modules that are defined previously elsewhere and imported into this code and actually into the E. coli code block. Here's a petri dish containing a gradient of chemoattractant, glucose, and E. coli. By executing this code, one should be able to see E. coli finding its way up the glucose gradient in a random walk fashion. I'll play the pages. Good. And L++ also applies to epidemiological modeling, such as for COVID-19, we can categorize, in this case, human populations into various states, and definition of reinfection cycles like this. And buildings like office spaces and single-family homes can be defined as an object in the system with their dimensions and access patterns during and outside of work hours can be modified. And furthermore, the maximum occupancy even can be controlled by specifying the highest number of people allowed in these buildings. And to model a global pandemic scenario, we can also represent the Earth and designate specific locations within it using GPS. And by incorporating detailed molecular infection models, we can even explore the possibilities for visualization and analysis at a higher resolution. Due to time constraints, I provided a brief overview of L++ syntax and some code examples. We're also in the process of developing extensive educational materials, including L++ book, detailed language manual on our website, and manuscripts that cover the entire breadth of biology programming technology. So stay tuned. And next, the Human Code Project. L++ stands at the forefront of the ambitious Human Code Project, envisioned as a groundbreaking successor to the Human Genome Project that we know that was completed in 2001. And our goal is to create a human interpretable code of human DNA, enabling the creation of an in silico human model. It'll become an integral tool across research laboratories and clinical settings, enhancing our understanding of human biology and investigating and debugging a myriad of human diseases. Current drug and vaccine development processes, hindered by lengthy and costly trials, will be transformed by the Human Code. Virtual clinical trials, enabled by this innovation, promise to expedite the development not only the development of drug and vaccines, but enhance scalability and reduce costs of clinical trials, all while minimizing human testing risk. And the human code promises a new era in personalized medicine. By adjusting specific segments of the reference human code, we can generate a digital twin of ourselves, the very specific and unique ones. And these models could predict the disease risk and help tailor preventative and therapeutic strategies and treatment strategies, marking a significant leap towards truly individualized healthcare for each patient and each person. And the complete version of the human code is estimated to be equivalent to 100 pages, 100 books, or about 100,000 pages. To accelerate the development of this human code, we plan to initiate and lead the Human Code Project Consortium, and this collective effort will gather hundreds and thousands of biologists worldwide to collaboratively write and experimentally validate and clinically test the code through regular meetings and various community forums. And now, lastly, I'll end with a brief discussion of L++ relevance in the era of AI. As we navigate the AI revolution, its potential to decode complex biological data is undeniable. However, the practicality of relying solely on AI for a comprehensive biological understanding is, I would say, because precise programming languages like L++ exist, and combined with AI's pattern recognition capabilities, I think they together offer a very complementary strategies for representing knowns and unknowns in all their aspects in biological systems. Here are some analogies I would like to make. In arithmetic operations, from simple addition to complex tasks, to more complex tasks, are efficiently and accurately executed by arithmetic circuits. In contrast, machine learning models fall short in terms of energy efficiency, speed, and importantly, accuracy for a slight probability of guessing wrong. And this analogy extends to operating systems as well, with a clear understanding of the desired functionality and behavior of the system. Programming languages enable us to precisely dictate an operating system's operations and task management. And relying on machine learning models based AI for representing this type of system would result in decreased energy efficiency, slower performance, and reduced accuracy again. And now consider biological systems like organs. Our knowledge is actually, compared to these two, extensively yet incomplete, with many aspects still to be understood. Here, a precise programming language like L++ can represent the known facets of an organism, and perhaps ML, machine learning models, can infer the unknowns effectively. Together, they again will complement each other and offer a comprehensive representation of a biological system. We anticipate that the initial human code drafted at the beginning to be hybrid of 50% L++ code and 50% AI models. Over time, as our understanding deepens about the system, that code will increasingly encapsulate our biological knowledge even more accurately and efficiently, aiming to eventually supersede AI models for more efficient and interpretable representation of human biology. Echoing industry leaders like Jensen Huang, we're approaching a digital biology revolution. With L++ and our research, we aim to contribute meaningfully to this transformative era, heralding new advancements in how we understand, interact with, and manipulate biological systems. So with that, I would like to thank Dr. Daewon Kim, the leader of the team, and the inventor of N++, Chani Park, for all of his software advice and the funding agencies that's supporting the current N++ team. Thank you, and I would like to take your questions.
Fantastic. So I amazed again. I mean, this is really amazing. So let me start with some question I still have, even though I asked many questions before. So right now, I sort of very optimistic, or I hope, in the future we have many, AI tool, programming, language, or biology programming, whatever, will be available in the future. But one thing, I also a little bit concerned about is there are too many other tools out there, and then too many database and data out there, but integration of entire AI tools and also database together, that might be very huge task. And also, if that all the database or the AI tool kind of separate in a sense, as a user, because I mean, we just wanted to use those kind of tool, but as a user, if that's kind of inconveniently kind of distributed to entire, local area with the limited access, that might be a little bit challenging. So I'm not sure you have thought about this kind of integration problem or not. I believe what you want to do is sort of integration with one program language, but we have the too many other databases, out there, and then we still wanted to use those things as well. So could you kind of elaborate how your program kind of integrate those things, and then how to integrate entire database out there, not only human database, but also bacteria, etc. It's my kind of long question, but yeah.
I see that it's got like two parts to it at least. One, the pure integration problem. I think all these like databases and data out there, they're acquired from different settings, that it's inherently just difficult to integrate data together because they don't work. They weren't in the same context. So I think it's actually the programmer's job to integrate those data into their code, L++ code, so that L++ code actually works. Whatever they wrote actually works with the data they integrated. So, and that can be done very modularly for each pieces, and then the way we can further integrate those together, these shouldn't have a problem because it itself is unit tested, the whatever code you have generated. The other thing is the availability of all these multiple databases, and people there's just so many things out there. Will they use L++ versus whether they want to use those other tools, that kind of thing. I think we just need to prove ourselves that L++ is the way to represent all biological knowledge, and then convince the users that you can really go much further by using this platform than others. And that's just pure the system design that we have to do, put in place so that it is intuitive for them to use, but also very powerful in what it can do once they start using it. So there comes with the visualization support of any system they write, also being able to debug the system by executing and stopping simulation, see where the problem is, and making logical and reasonable inferences as to like where you can find potential problems in your systems and so forth. So with all of these kind of tools that come with L++, it's going to be a very instrumental tool for every biologist. So we actually hope to take advantage of all these data that's out there and let biologists think about that and see if that actually makes sense to put into a single program. So that's kind of like combining the two questions I try to answer to.
Yeah. That's fascinating. The very long question and but elegant answer to my question. So the reason I asked that question is there are some discussion among government people and industry and also academia people how to integrate gigantic amount of data and then all the database and also all the tool we will have. The problem in biology, as biology is messy. Biology is very complex, also messy. But at the same time, biological researchers, including myself, somehow develop things individually, separately, rather than one coherent or consistent way. I'll give some examples. For example, we still have two computer operating system, Mac system or PC system, Windows. They are not compatible, but we only have two, for example. But in biology research field, we have too many. I mean, we have one protein prediction program, but we have another. We have RNA prediction program. We have another, and we have too many other database, and they are not talking each other because we did not systematically develop things based on PC compatible or Mac compatible. We just develop everything just alone without talking to other people because we don't have any standard. We don't have a standard kind of thing. And then everything is just, separately developed. So that might be, challenging in biology. That's why, still biology is so complex. So describing is also, much harder than, just computer programming. So that's why, I'm talking about this kind of problem. So having said that, I have one question, so I want to address this one first. So one question actually coming from audience, before I just ask one other question. So question is, how does L++ handle interactions that usually require specialized programs like protein-protein interactions and ligand docking?
So I think that's the resolution related question. So L++ support the description of super high resolution interactions like at atom level interaction, as well as even lower resolution or high level processes using much higher level interface like molecular interactions using kinetic dissociation constants. And so there are reaction models that we support at various levels of resolution. So that really depends on the user. If they wanted to use that level of description, they can. We just need to support that, provide a syntax for it, and be able to facilitate it. And it's the same problem actually going up as well. Like, how do we describe the function of an organ as well? So, but we have ways and universal ways to handle all of that using reaction syntax and providing appropriate models that handle all of those. So how we simulate that is like another sort of a question, right? That itself is another technology and technique. So I won't really go into detail there. But as long as we provide a ways to describe whatever the user want to do or describe at the resolution they want, that should all be provided with the language.
Okay, wonderful.
Yes.
So my follow-up question is a really crazy idea when I listened to what you described earlier and also today. So one day, what I want to, probably not within my time of life, but probably my daughter's generation. Or one day I want to see, we basically do our human genome sequencing for individual people. And then let's say the all entire sequencing and then also done some RNA sequencing as well. And then whatever phenotyping, everything done. And then that all into the whatever programming using your, ARPA-H. And then one thing I want
Human
to,
code, yes.
yeah, one thing I want to see in the future is somehow that program somehow embedded in my arm or the wrist. And then
Okay.
if I have some, I don't know, some food kind of, or some pollution toxic compound somehow exposed to that one, I want to calculate what is my, risk fact, level because
Yeah.
of that, and then how to cure or,
Give you
neutralize
suggestion.
that toxin and
Yeah.
just immediately. And then, is it possible, or this is part of your potential project down the road? I'm just curious.
Yeah, I mean, thinking what the language can describe,
Uh-huh.
100%, I would say. I think it's a matter of creating syntax to describe things that's needed to do that, right? And then also supported by the rapid simulation technology and the user interface that's able to communicate between the user and your simulation. So yeah, I mean, and feeding the data collected about you into the reference human code and being able to make minor changes, right? We're like 99% the same in terms of change. and other types of data, like your blood work, or it could be your RNA-seq data at one certain time point. Yeah, I think all of that can be easily integrated using this kind of programming language. And I think it's the very technology that one may pursue, actually, because yeah, like you said, it's like an immediate follow-up sort of thing I want about ourselves if we were able to simulate it, right? Describe everything and simulate it appropriately and be able to predict. And now with even AI, give suggestions on how you may adjust your behavior to, um, live longer, live healthier, and lose, I don't know, extra weight. Keep your mental health intact and all of those things. Yes.
Okay, that's wonderful. So I'm now seeing the movie coming, live, movie. So the follow-up question, probably last question I have. So the reason I asked that question is, I believe, potentially accomplishing making the whatever program you wanted to make, that might be possible. But real challenge would be if we wanted to implement what I just suggest, that require personalized programming, and then to basically achieve the mass production of that personalized programming, that will be very challenges.
Yeah, I don't. So once we have the reference human code, there won't be as much personalized programming because you can use most of the reference human code and be able to just edit the parts that's different about you from the reference. So I don't think there's going to be as much personalized programming. I think there will be actually a lot more work initially to build that reference human code, and everybody can just use it as a template.
So you are saying based on one human's genome sequence and then make the program.
Just the general human biology
Yeah.
from, let's say, a reference human genome or a particular person, sure, somewhat of a healthy person without, say, noticeable like diseases or something like that, genetic diseases. Yeah, sure. You can take that as a reference, and then whatever differences that you can spot, then you can replace that code or overwrite the relevant code, just so that you have your own version for that part, and rest of it is not going to be much different in how it behaves. Anyway.
Okay, wonderful. Now, now I have one more motivation to live longer, to see that
Please.
that really happening, in my life. That's
Okay.
wonderful. Okay, so let me check. Okay, we don't have any additional question from audience. So let me close because we only have 2 minutes left for the 11 o'clock my time. All right, so amazing talk again. I really fascinating. Okay, so thank you all for joining and staying today. We'll meet again on February 29th, the last day of February. Also only happening every other 4 years, I guess. The Thursday and same time and same Zoom link. We'll have the Professor Beatrix Sussis at TU Darmstadt, Germany, and also Dr. Sri Sankaran at the Leipzig Institute for New Materials, also Germany. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. Just 1 second.